Good evening. Welcome to our session on genetic therapies. We're excited to talk to you about various aspects of genetic therapies for CF, and importantly, we've dedicated a block of time to take your questions in a live Q&A at the end of the session, so please submit them. My name is Kathryn Singh, and I'm a professor in the School of Medicine at the University of California, Irvine, and I'm also the genetic counselor on the PED CF team at Miller Children's Hospital. I'm joined tonight by three excellent speakers: Elyse Jupiter, Dr. Michael Harnish, and Dr. Dan Rorgan.
Hi, everyone. I'm very excited to be here tonight. I am Mike Harnish. I'm a rare disease researcher. I've also worked in a lot of other areas of the healthcare ecosystem, and I'm a CF patient myself. And again, excited to be here. And Dr. Rorgan, would you like to introduce yourself?
Hi, everybody. I'm Dan Rorgan. I'm one of the docs with the Adult Cystic Fibrosis program at the University of Pennsylvania. I'm excited to be here too. Thanks so much for everybody who's attending. And I'll hand it over to Elyse.
Hi, everybody. I'm Elyse Jupiter, mother to Tobias Jupiter, CF parent of a child with a rare mutation. Tobias is actually the only CF patient in the database with his mutation combination. Which means he doesn't qualify for the current modulator therapies that everybody is, familiar with. And so tonight we're going to talk more about the genetic therapies that I'm very interested in, because we're still looking for our definition of hope. Right in the beginning of this journey, we were thrown right into the dark side of CF. The long NICU stay, multiple surgeries, all the specialists, all the complications. By 6 months old, he was doing all the therapies that a lot of CF patients aren't doing until they're 4. So, we've been very aggressive with his treatment, but, it's just been kind of a constant barrage. And in the beginning, we had so much hope for all the things that were immediately available, but we started to learn that, he just wasn't going to have those opportunities because of his rare mutations. This old traditional CF path is really scary, watching your child decline and throwing the kitchen sink at it, and just, hoping that you can kind of keep things good enough while you're waiting for the real game changer. So the genetic therapies offer, lung saving and life extending opportunities, buying time and preserving the quality of life while still hoping for a cure. We managed to throw enough at it that he went 4.5 years without a hospitalization. But about a year ago, he had his hospitalization for an exacerbation for the first time. And that was like a whole different world than the last time we'd been in the hospital. He was the only CF patient inpatient at the time. And that is just a huge testament to the benefit that the modulator therapies have on the CF population. They used to have a dedicated CF respiratory team at the hospital, and they were gone. There were just a few left over that even knew the protocol for the breathing treatments for CF, which is just a huge difference from 4 years ago. So, we've seen the success in the majority of the CF population, but there are so many things in the pipeline that we're going to talk about later tonight. Just in a few minutes, we'll get into the details of it, that are, they're everything to my family in terms of real opportunities to change the trajectory, so Tobias can have more days playing soccer than he does doing treatments. So with that, I give it back to Kathryn Singh to talk more specifically about those therapies.
Thank you, Elyse. So Mike and I are going to give you an introduction to genetic therapies in CF. And a reminder that we have Q&A time at the end. So please, you can submit your questions as we're speaking, and then we'll cover them at the end. And these are disclosure slides. So we're talking about genetic therapies. So that means we have to talk about genes. And I imagine for this audience, most of you are very familiar with genes. But just a reminder, genes are our body's instructions to make proteins, and they determine how our bodies develop, how we look, and how our bodies function. And the gene that's involved in CF is called CFTR. And we have, excuse me, the CFTR gene, along with all of our other genes, provides the instruction to make this intermediary called RNA that actually is the template to produce the protein. And in the case of CF, the job of that protein is to move ions, both chloride and bicarbonate ions, across cell membranes. And that's shown, the chloride ions are shown in these little purple circles here. And we actually have 2 copies of almost all of our genes, and that includes the CF gene, where we get 1 copy from mom and 1 copy from dad. And because CF is autosomal recessive, that means if we have a mutation or a disease-causing variant in one of those copies of our gene, so that we are not producing protein from that copy of that gene, we are protected from having CF because we have that backup copy. But if we have a mutation in the other copy as well, such that we produce no CF protein, then CF occurs. And all of the pictures that you're going to see for the rest of the night focus on 1 copy of DNA, but remember that there really are 2 copies. So again, that process of going from the gene to the protein, it's called the central dogma. And it's the process of copying the DNA into RNA, which is used as the template for the protein. And on the previous slide, I showed you that when there's a mutation in the DNA, you're not producing CF protein. And that's the case much of the time. But there are various types of mutations in the CF gene that lead to different effects on the protein. So for example, you might have a mutation that still produces a protein. That protein just may not work as well as we want it to, and it may not be moving ions in the same function that we want it to. Or we might have a mutation that causes, sorry, I went too fast here, no protein at all. Or we might have one, and my slides got messed up, but there was one in there somewhere where you just produce protein, but not enough of it. And then there's another example where you're producing an RNA, but it's an altered RNA. So that RNA can't produce the protein that it's intended to do. And so now we're going to look at these concepts actually in the cell. So you can imagine that we're looking inside of a cell that's lining the lungs. And right now we're looking at an example where there is no mutation. The DNA is intact. So we have a correct copy of the CFTR gene. So again, where we were going from left to right before, we're going to go from bottom to top. So that DNA produces an RNA, that's the intermediary to build a protein. And the protein gets processed and finished, and it moves up through the cell, and it sits on the surface where it moves chloride and bicarbonate molecules from inside of the cell to outside of the cell. Now we're going to advance this a little bit and show what happens when you have a mutation. And there's actually 5 different classes of mutations, and we'll look at them one by one. In this category, there's a mutation that affects the RNA. So instead of producing this blue full-length RNA, we're producing a shortened red RNA. And that abnormal RNA cannot produce a protein, so you get nothing up here on the surface to move the molecules. And then there are other varying degrees of mutations that have different effects. So you can produce a premature protein that doesn't get completed and doesn't last to survive up to the surface of the cell. Or you can have mutations that produce a full protein, but it doesn't function the way that it should. And the process of moving those chloride molecules is not effective. Or you have a completely normal protein, but there's just not enough of it. And that happens because of something called splicing, where you're producing different types of RNA. If you make the right kind of RNA, you're making the right kind of protein. But if you produce the incorrect RNA, it's not going to make a protein. So your net effect is not enough. And so a lot of the focus that has been in the last few years is on CFTR modulators. And what CFTR modulators do is they focus on the protein, either a very early stage protein or a later protein that's not working or is in an insufficient quantity. And by giving the modulator, it increases the transport of chloride across the membrane. So you're maximizing what you can get out of the effect of that protein. The problem is, you can see in this first category here, there's no protein to modulate. So that's why modulators don't work for people with mutations in this category. These are the nonsense or the truncating mutations. And in fact, there are mutations in each category where modulators don't work. And some people with these mutations are not eligible for modulators either because they don't work, or maybe they're so rare that they've never been studied and we don't know if they work, or they may be genetically eligible but can't take modulators for a variety of other reasons. And we know that overall in the US, about 90% of people have a mutation that is eligible for a modulator from a genetic standpoint. But unfortunately, that 90% doesn't translate when we break the population out by different racial and ethnic groups, as Dr. McCauley and Dr. McGarry showed us in 2021. And I actually like to flip these numbers so you can see the percentage that don't have access to a modulator based on their genetic mutation. And while overall 10% of the US population doesn't have access to a modulator based on their mutation, if we look at non-Hispanic whites, it's actually less than 10%. And concerningly, in Black and African American people with CF, it's 3 times that at 30% without access to a modulator based on their mutations. About 25% of Hispanic people with CF, and about 20% of people of other races with CF. And this is clearly a problem. So if modulators aren't the solution for everyone with CF, we refocus from targeting the protein, like we've done with modulators, to targeting the DNA and the RNA. And you can see that those are present in all the categories here of people with CF. And this is where thinking about genetic therapies and research on genetic therapies becomes important in order to get treatment to everybody who has CF. And when these genetic therapies enter trials, actually, they're already entering trials, but as we build our trials more and more, the early stages will target people who don't have access to modulators. So a big population from this category, but also people from all of these other categories who may not be eligible for modulators currently. And Mike is going to talk a lot more about what those therapies actually look like and how they work. But before we get there, I want to talk a little bit about why this research on genetic therapies is unique from therapies that we're used to talking about and studying in CF. And so the first is that genetic concepts can be unfamiliar and sometimes a little bit intimidating to people who aren't used to talking about them. I'm sure until tonight, many of you hadn't heard about the term central dogma in terms of talking about DNA to RNA to protein. And if this isn't something that you're doing every day, this isn't going to come second nature. It's also a hot topic, especially mRNA, in the media and in politics as a result of mRNA being the active therapy for preventing COVID-19 in terms of vaccinations in this country. And because of that, people have become more familiar, for better or worse, with mRNA therapies. And our DNA is what makes us unique. We are all uniquely individual because of variants within our DNA. And our DNA makes us who we are. And so there are inherent privacy concerns when we think about studying DNA or targeting DNA in terms of research studies. There's also a real fear of discrimination or being taken advantage of by researchers. And these are real concerns because they're based on a history of unethical and, in some cases, abhorrent research practices that happen in this country. And while we've come a long way since the time of those research studies, the stains remain and the fears remain. And so these, as well as many other concerns that people may have, are things that you'll want to discuss with your CF care teams and with potential research teams as you consider participating in a genetic therapy trial. So to go back to the first point, I'm going to hand it over to Mike, who's going to talk more about how these therapies will actually work.
Thank you so much, Katie. As she said, I'm going to be speaking a little bit more about what these therapies are, how these actually work. My background is I was a rare disease researcher for several years, and I now work in all sorts of different areas of healthcare. I'm also a CF patient, and I've been giving talks with the Cystic Fibrosis Foundation for a couple years. I actually love that you can see me on the screen because I got used to talking in front of crowds. I use my hands a lot, so this is absolutely fantastic. I'm very excited. So to really understand how genetic therapies work, I'd like to touch back on this central dogma that Katie talked about a little bit. So how you get from DNA to protein. So inside all of our cells, we have DNA in a smaller compartment called the nucleus. Now, what happens is this DNA gets copied into sort of a one-time set of instructions called RNA and shipped out into the cell. Once that RNA is shipped out into the cell, it acts as that set of instructions for the cell to make a protein. And once we have that protein, it moves to where it needs to move in the cell and carries out its normal function. or at least it should. For people with CF, of course, we have mutations that prevent this normal function from occurring. So there are a couple ways that we can use genetics to correct this. The three that I'm going to talk about today are mRNA, gene delivery, and gene editing. And how each of these work is basically mRNA therapy is providing the one-use set of instructions for the proper CF protein. Gene delivery would be bringing in a permanent DNA copy of the correct CF gene. And then gene editing is actually going in and correcting the error that exists within the CF gene, so we make normal CF protein. The goal for all of these is the same, though. We want to get to a functioning CFTR protein. So to think about this sort of as an analogy, think of the case where CFTR is a car and something has happened to damage the car, but you still need to get around. How could you solve this problem? Well, you could go ahead and rent a car or use a rideshare, a taxi, and that's sort of what the mRNA is. It's this temporary one-time use as you need it. You could also think about just buying a new car. You'd still have your old car, it would still be broken down, but you'd have this new car and you could use it to get around. That would be gene delivery. And finally, you'd have gene editing, which is where you just repair your current car and use that to get around. So at a high philosophical level, this is how these three types of therapy work. Now that you have the idea of the central dogma and the philosophy of how these work, I want to get a little into the technical aspects of, okay, but what's actually going on with these therapies? And the first one I'd like to talk about is mRNA therapies. So the idea with mRNA is we deliver genetic material using a virus. And I'm going to stop right there because virus usually sends red flags up in people's mind. But really, viruses are just nature's syringes. We associate them with disease because in the wild, the viruses that make us sick contain DNA and RNA that causes our cells to do things that is harmful to them. But just like how a syringe could be filled with poison or with medicine, viruses themselves aren't harmful. If we put RNA or DNA into them that we're interested in having in our cells, like in the case of genetic therapies, they are very helpful and very convenient. Viral vectors have been used in this way for genetic research for decades, and there are gene therapies already on the market, prescribed, treating people that use these viral vectors. This is not a new technology. This is not a dangerous technology. Just want to put that out there. All right, so now that we all love and appreciate viruses for their contribution to genetics, let's talk more about this mRNA and how it works. So the idea with the mRNA treatments is we have the viral vector. It's delivering this RNA, which is the one-time use instructions, into the cells. The cells are using that proper instruction to make a proper CFTR, which is going to do all the things that CFTR should. Because this is RNA, because they're one-time use instructions, this isn't sort of a one and done permanent solution. This is something that you'd have to continue taking. If we want to think about the more permanent solutions, we can think about gene delivery. Now, gene delivery is a bit different because it's actually using DNA instead of RNA. So rather than putting in these one-time instructions, we're taking a CFTR gene, putting it into the cell, and it will make its way into the nucleus where DNA lives. Now, inside the nucleus, you still have your old mutated copy of CFTR, but you also have the proper copy of CFTR. So when you go through this process of making RNA, sending it out, translating into protein, sure, you're still making the CF version, but you're also making a proper version of the CFTR. And just like Katie said, this is an autosomal recessive disorder, meaning that if you even have one copy of the proper CFTR, you will not have CF. The next technology is gene editing. So gene editing is a little different in that it's not providing one-time RNA. It's not providing a new copy of CFTR. Rather, this is putting in molecules that edit specific sequences of your DNA. And the goal with this technology would be to edit the mutation in our CFTR gene to make it the wild type or healthy version. Once this editing is done, for all intents and purposes, we no longer have a mutated CFTR. All the RNA we make is correct, all the protein we make is correct, and it functions properly. Now, I think the most exciting thing about these technologies is they sound very, at least to my ears, they sound very science fiction, very far off into the future. But medicines using all three of these technologies actually already exist and are being used to treat different conditions. As Katie alluded to earlier, I think a lot of people are familiar with RNA because it was the basis of a lot of the early COVID vaccines in the US. However, gene editing, there's actually a rather new therapy to treat sickle cell anemia that just came out a few months ago. And gene replacement, where you put in a functioning copy of a gene. I just have one disease listed here, but there are several diseases where gene replacement is used to treat a disease. Now, I want to take just a moment to talk about one of the big concerns that I hear people have with gene therapies, and that is really fertility. Will this affect my fertility? How will this gene therapy affect my children? And the short answer is it won't. The gene therapies I've talked about are called somatic gene therapies, which you can see on the right side of the screen. Somatic cells are sort of your adult differentiated, we call them cells that have a job. Think your lung cells, your nerve cells, things like that. Somatic gene therapies specifically target certain cell types, certain genes, and cause changes in just those. And that has some positives and negatives that we can talk about later, especially with CF, where it can affect multiple tissues in the body. But it doesn't affect the sperm and egg cells, which are, what controls fertility and ultimately what will give rise to a new human and all their genetics. The issues with fertility and if you'll pass it on, and a lot of bioethics questions as well, are the realm of germline gene therapy, which again is not what any of the ones I've talked about are. Germline gene therapy means editing to either the sperm or egg cells or the very early embryo. And because our entire body, is a result of these genes, these cells, germline gene therapy will carry forward in the population. That's the one that has more bioethics discussions about it, more regulation discussions about it. But again, that is not what the therapies that I've talked about are. That is entirely separate. The therapies I've talked about are somatic gene therapies. They do not affect fertility, and they are not heritable to the next generation. Now, this certainly isn't all the bioethics and safety questions that we'd like to cover. But to go over those, I'd actually love to hand the presentation over to my colleague, Dan, who will go through those in more detail.
All right, thanks, Mike. Yeah, so Mike started talking about some of the ideas about concerns that people have related to genetic-based therapies. And so I'll be talking about those details a little bit more in the next 15 minutes. Just for disclosures, I do receive research funding for studies in CF, including for genetic-based therapies, but I won't be talking about any of those specific studies in detail tonight. So breaking things down into broad categories, and again, just as a reminder that we'll have a Q&A session, so if people have more specific questions, with limited time, we're just going kind of broad brushstrokes, but we'd love it if people give us more direct questions of the things that are really a focus of your mind. But so the broad categories I'm going to talk about is, what I'm going to spend the most time on is talking about safety of the treatments themselves. I'll touch a little bit more on the fertility and future generations, just a little different twist on some of the things Mike was just talking about. And then there's the third part, which is, let's say everything goes great. We have a research program that shows a treatment that's incredibly effective and gets approved by the FDA and then is available for people to use outside of research studies years from now. There's that question of what will it cost and will everybody have access to it who needs access? So when we're talking about safety, I think the real question isn't so much, can we guarantee 100% safety? Because the reality is there's no way to guarantee 100% safety of anything. Even medications that are approved by the FDA and that you can order through a pharmacy. We all know that people have side effects to medications that exist. So the real question here is trying to avoid serious safety concerns, things that are more life-threatening or have severe impacts on somebody's overall health. And so then the real question is, well, who's keeping an eye out for safety to make sure when somebody's going into a research study, they're not a guinea pig? Katie referred earlier to some of the horrible history of unethical research in the United States, and part of that is because people were being put through research without being told that they were being put through research. And that's some of the changes she's talking about, is now there's so many layers of monitoring to make sure nothing like that happens. So looking at who does that at different levels, so starting at a national and international level, governments play a big role in this. So the Food and Drug Administration, or FDA in the United States, the European Medicines Agency, which is also called the EMA, are the two the biggest in the world, but many countries have their own regulatory agencies that can actually completely shut down a company's ability to move further with research. If they have concerns about safety, then the government can say research stops on this compound that is showing safety concerns. We also have in the United States the Cystic Fibrosis Foundation, and then there are other advocacy organizations for cystic fibrosis in other countries that pays a lot of attention to the design of research studies for people with CF. A couple arms of the Cystic Fibrosis Foundation that play a really key role in this. There's the Therapeutics Development Network, which is a combination of many cystic fibrosis centers throughout the country. And this involves review of any research protocol to go through a lot of details. One is this protocol realistic? Can it be accomplished? Does it look like this is something that has enough possibility of working that it's worth studying? And then, of course, the safety aspect. Are we actually doing something that's safe here? And then there's the Data Safety Monitoring Board of the Cystic Fibrosis Foundation, and that is specifically focused on the safety of treatments that are being used in research, and not just in the design of the studies, but also monitoring as a study is ongoing. So if any concerns arise during a study, again, that they can play a big role in putting the brakes on a study moving forward. A key point with both the TDN and the DSMB is they have representatives from the CF community. So it's not just researchers and pharmaceutical companies and things like that. Well, pharmaceutical companies aren't actually part of these within the CF Foundation, but it's not just CF researchers and clinicians, but there are also adults with cystic fibrosis or parents of children with cystic fibrosis on all of these committees to have the input of people who are living with CF on a day-to-day basis. This slide is intentionally complicated and not for people to look at in detail, but just to show when the TDN and the DSMB are looking at research, they're going over things with a fine-tooth comb, looking at every possible detail that they can. And there are many people that are part of this process. So just seeing that behind the scenes, it's not just that there are these names for these arms of the CF Foundation, but that they're taking this very regimented stepwise approach to make sure that research is being assessed for safety as we go along. And then at the local level, every institution that's participating in a research study has to submit the protocol to an institutional review board. So that's another set of eyes that's looking at a study to determine that things are being done safely. When we're talking about genetic-based therapies, there are institutional biosafety committees, which are generally connected somewhat to the institutional review boards, but these are people who have a specific expertise in genetic-based therapies, so they can raise any concerns that they can identify with their expertise that other people without the same expertise might miss. And then, of course, there are your CF care teams. So when we're working with people who have cystic fibrosis, and these are people that we care about and interact with on a day-to-day basis. And so as a physician taking care of people with CF, it's important to me not to put the people I'm working with at risk unnecessarily. And I think anybody who's dealing with CF, you have a care team that feels the same way about you. And so the people who are involved in the research protocol are CF care teams themselves. And so that might be your own team that's participating in the research study, or you might be getting referred to another CF care team, in which case you have two CF care teams looking at things. So again, just more sets of eyes that are assessing the protocols to make sure that we're not putting people at unnecessary risk as research is pursued. So in terms of the specific things we do to protect safety, so of course, the first bullet point there is just everything I've been talking about already, just all the many layers of people looking at studies. But then there's things like preclinical testing of new treatments, which is, by preclinical is just a term for all of the research that's done before a single human being receives a treatment in research. So there's a lot of lab study that goes on that's looking at cells in a petri dish. And then there's animal testing. And granted, there are some people who are uncomfortable with the idea of animal testing. But the reality is if we don't test in animals, then the first human who's getting a treatment would then be getting it with just less information about what sort of safety we're dealing with. And so the animal testing of anything that's being used for research purposes really provides that extra layer of safety, that if there are safety concerns that come up in animal testing, then that treatment can be shut down before it ever is given to a single human being. There's also slow enrollment of these studies. So if you think about something like a medication for high blood pressure that's being studied, that might be given to thousands of people in a big research study, and they're trying to enroll people very quickly. But this is very different, since this is something new that we're trying to assess safety very carefully. The studies generally enroll 2 or 3 people, and then a pause so the Data Safety Monitoring Board can look at what have we seen from those first few people who participated. If everything looks okay, then move forward to the next few. If there are safety concerns, then that will alter how things are done with the research going forward. So it's not that all of a sudden multiple people are being exposed to the same treatment. As we're learning about this, we try to do it slowly so we can do it safely. There's also long follow-up, particularly with things, Mike talked about the different types of treatments that have different durations that we would expect them to last, with the DNA or gene editing that would potentially last for a long time. And so then we're not just following up people for days and weeks after getting treatments and research studies, but it's really months and years when it's these genetic-based therapies to see what are the long-term effects. And initially, when studies are done in people, it only includes adults, which actually for some parents of people of children with CF might feel a little impatient hearing that, but part of it is just the idea that we want anybody who's participating in these studies to be somebody who can actually consent for themselves, go through a very detailed informed consent process, and make this decision on their own behalf. And then if we see safety and effectiveness in adults, then those studies do start to move through towards children as well, as we've seen with a lot of the modulator therapies that started with adults and 12 and older, and then went to age 6 to 12, and so on to younger and younger kids. And so here's an example. The CF Foundation website, cff.org, has a ton of useful information, including this drug development pipeline, which just shows what are the different drugs or treatments that are in the pipeline to be developed as possible future therapies. And so I've highlighted this preclinical section, which just shows everything starts in preclinical. And you can see a lot of these compounds here under genetic therapies are still in the preclinical section. Some of them may make it to people with CF in research studies, and some may have safety concerns where they never make it to any human being. And then you can see some of the others are into phase 1, which is the early phase of giving it to people with CF, or even to phase 2. And actually, and some of them, phase 1 actually starts with healthy volunteers before they go to people with CF. But again, this is just showing that there's that robust process of looking at things in settings before giving it to people. And so in those few studies that have actually given genetic-based therapies to people with CF so far, again, there haven't been a lot of people so far who've received these treatments, but the things we've seen, some people have gotten fevers, which generally get better in a day or two. There have been people with pulmonary exacerbations of CF, which of course can happen outside of receiving genetic-based therapies as well. There's one person in one study who had severe lung inflammation that did improve. and was very closely assessed by the Data Safety Monitoring Board to make determinations about what to do next with that. And then with any research study, there's always a possibility of side effects that we haven't learned about yet. If you have something that's a 1 in 1,000 side effect, and so far 100 people have received something, then we just might not have seen that side effect so far. But when we're studying any new treatment, the real key point here is that safety and side effects are actually what comes first. We want things to be effective, but before effectiveness, safety is really our top priority. And then the other part is, Mike talked about some of the treatments that are in existence now for spinal muscular atrophy and sickle cell disease. And then there's just research in many other medical conditions looking at genetic-based therapies. So we can learn from that. If there's a safety concern that comes up with respect to a specific technology that's being used for another medical condition, we can learn from that and modify plans for research in CF to avoid things that are unsafe. And then Mike talked about the viruses. So I'm not going to talk too much in much detail about this, but again, it is something that can seem weird. Like, why are you intentionally giving me a virus? And I think Mike did a really good job explaining that viruses are just very effective at getting things into cells. And I borrowed this slide from Kara Fauché from the CF Foundation. I think she's brilliant at the way she communicates some of these details. And she brought up this analogy that's showing the idea that, to us, DNA and RNA is incredibly small, but from the level of the cell, not all molecules are the same size. So Trikafta compared to CFTR mRNA is a dramatically different size. So it's like comparing the size of a kumquat to a watermelon. So Trikafta can get into cells pretty easily, but something as big as CFTR RNA or DNA is just too big to get into the cell by itself. So it needs help. Viruses are what are studied the most for genetic-based therapies. There's also something called liposomes that are being used as another vector, and vector is just a term for a delivery device to get something inside a cell. So since you need to get these into cells and they're big, it's just we need help from something else to get them into the cell. But then the flip side of that is also, that's one of the other steps of safety that ties in with what Mike was talking about with fertility, is these big molecules are hard to get into the cells that we're trying to get them into. And that's why we're actually starting with the lungs, where it's using these treatments with inhalation to get to cells of the lung. And then it's hard to get past that point. And so we don't expect it to get past that point, which is good and bad. It's good from a safety standpoint that we can be comfortable that it's not going to have widespread effects beyond the lung. But it's a downside from the standpoint of, if you're looking for a beneficial treatment that's going beyond the lung, well, we're not going to get that with the early stages of these genetic-based therapies. And so again, that ties into the fertility and impact on children, is we're not expecting these initial treatments to get past the lung significantly. So they should not get to the sperm, should not get to eggs, so shouldn't have any impact on fertility, shouldn't have any impact on future generations. Now, there may be something 10 years, 15 years from now that is something that's more body-wide, and that's where, with all these layers of monitoring for safety, there will be very close attention paid to those kind of things. But these early genetic-based therapies are strictly focused on the lungs. And then other unique challenges related to genetic-based therapies. One thing that people who have participated in research before, something that will be unusual about this is, with most research studies, you've been in one research study, you can then be in as many more research studies as you're eligible for. But when it comes to genetic-based therapies, having been in a prior genetic-based therapy study might actually make you ineligible for the next genetic-based therapy study. And part of this is depending on what type of treatment and just the way a specific research protocol is written. So Mike talked about how RNA is more like the rental car. It's a short-lived molecule. It breaks down very quickly and then goes away, which then means it would also need to be dosed repeatedly. So something like that might not have as much of an impact on eligibility for future studies. But again, each study defines its own eligibility criteria. So a study could say, yeah, even with prior RNA in research, you're ineligible for the next study. Whereas things like DNA, it's longer lasting. We don't know for certain how long lasting it's going to be. That's part of what we're doing with research. But then that might be something where a research study might say, well, now we don't know if we give you our genetic-based therapy in another research study, and we see something good or bad, is it because of the thing we just gave you now, or the thing you received in a research study a year or two ago? And so that's why it could impact your eligibility for future studies. And gene editing would be even a more extreme example of that, because if there's actually a fundamental change of the gene, then that could be something that really has a long-lasting or even permanent effect. And then there's the question of cost and access. And I think there's no way around this, that if people have read the headlines about the treatments for sickle cell disease, for spinal muscular atrophy, they're really expensive. We're talking about a treatment that costs more than $1 million. And of course, that's something that nobody is going to be paying for out of pocket. And so the real focus here is making these treatments, after we get through research and find really effective treatments, making sure they're accessible to everybody who needs them. Katie talked a lot about how we're dealing with the people who are not eligible for modulators are people who historically in the United States have not been given their fair share. And we don't want that to carry over to something like genetic-based therapies either. So the key here is the CF Foundation, particularly the Compass, which is part of the CF Foundation, advocates strongly for providing access to people for therapies. And the CF Foundation is sort of the model advocacy group. Other medical conditions kind of model themselves and say, hey, we need to be more like the CF Foundation in terms of how we approach things. And keep in mind, Compass actually applies not just to future things like genetic-based therapies, but if there's something now where you're having trouble getting the treatments that you need, Compass can be a great resource to turn to try to help get the things that you need. And then the other point about access is how soon. So we're talking about going slowly for safety, but then there's the flip side is, and I think Elyse has spoken about this when I've spoken with her one-on-one, is it's just, there's also that idea where we don't want to wait too long. We need these treatments now or as soon as we can possibly get them. And so it really is just, there's always this balance, trying to go slow enough that we can do things safely, but quickly enough that we can get treatments to people who need them. And one thing that can just really help move this forward so the treatments do become available is if you consider participating in research studies, then that's going to be what helps move treatments closer to the finish line where they become available for everybody. And there are a lot more resources on cff.org, including this specific page on genetic therapies. But you can find a ton of information about research, clinical care, and so much more on cff.org. So I encourage you to go there to look more, including if you're looking for a clinical trial to participate in, they have a clinical trial finder there as well. So I'm now going to hand the mic over to Elyse and Mike, and then we'll open up for Q&A with everybody else.
Thank you so much. Like Dan said, we're opening up Q&A, so please feel free to submit questions. But while we wait for that, I really just wanted to have a talk with Elyse, because interacting with CF patients now and parents of CF patients, both who may be eligible for modulators and who aren't, has really brought this idea of community in my head, because when I was growing up, everyone's CF is different, but we were all sort of in the same boat. Everyone was looking for the next treatment. And we all kind of assumed that the treatments would be for everyone. And again, everyone's journey is unique, but we were all kind of in the same boat. And that's changing a lot. Somebody like me who's grown up and has had their CF progress versus someone who's very young and eligible for modulators, someone who has a child who's very young but isn't eligible for modulators. We all have very different experiences, and I can look back on it, but Elyse, I'd like to know from your perspective what this journey looks like.
I mean, I don't know. Sorry, got a little brain freeze on me, but I'm really just excited for what the options are. We've been asking every month, every time we see the CF, is there anything we can sign up for? So the journey looks a lot like me attending this and making sure I stay on top of presentations so that, I know what to ask about, so that I can, learn what the choices are and what the risks are associated with it. But, from our perspective, there aren't any other choices. So we're just kind of sitting our hands a little bit, doing what we can in the meantime. I'd be curious to what your perspective is. that you're on a modulator therapy, but what your consideration might be for these other therapies that could be available to you.
Yeah, absolutely. So I am eligible for modulator therapies. I'm on modulators, but they aren't perfect. They aren't a cure. Some people have really severe side effects on these and have had to even come off of them. So as much as there is a difference where some people are eligible for modulators, others aren't, I still look at this research with a lot of excitement and hope, because they might be better. They might be more tolerable for these gene editing. If they are permanent, now we're starting to talk something that looks a lot like a cure on paper. So while there are different levels of stakes for people depending on what they're currently on, what they're eligible for, I think everyone with CF still has a stake in this research, still should be excited about this research, and still should stick together as a community in supporting all of this. Because first off, yes, it does benefit everyone. But second off, we don't want to leave a single patient behind, period.
Oh, absolutely. But I also, we don't know what the long-term trajectory looks like for any of these things because it's so new. So, I mean, exploring other options is essential to making sure that there continues to be pathways for everybody, because what if what you're doing stops working and you need to hop to something else because, the disease is so varied across everybody's experience.
Yeah, that's a great point. As something that works now might not work down the line. There's no guarantees. As Dan said, it takes a long time to see the efficacy of these sometimes. So, yeah, we're definitely still in it together. I think we just need to embrace that as a community and stick to our guns there. It does look like we have enough Q&A questions. So Elyse, thank you for having that discussion with me. But yeah, I think we're going to move to Q&A.
Okay, so our first question, what are some of the key concepts that patients should understand prior to making a decision about gene therapy? We can probably all contribute to that one. Dan, do you want to start?
Yeah, well, I think it's going to vary a lot depending on the specifics of the study. So because it's going to be important to know, is this a therapy that's considered something that will be short-lived, or is it something that we're anticipating a long-lived response, or potentially a permanent change? And just getting all that information about what do we know about the safety, and any consent process should involve this. Like what was seen when this was studied in animals. Has this been given to anybody without CF? Just as I said, sometimes they have healthy volunteers who are the first in line to try a new therapy. So just getting as much information as you can about what do we know about this specific treatment so far, so that you can make a decision about whether you want to be the one to participate in the next step of research.
I think timeline is also important. Some of these trials require very frequent dosing. And so if you don't live close to a center and would have to travel far, is this something where you need to be dosed multiple times a week or every few weeks? In terms of frequency of needing to come to visits and come to evaluations is another really big consideration. And another one that Dan talked about is, depending on the type of therapy, how might this affect eligibility for future therapies? If you're someone who's very interested in participating in multiple trials, understanding what the course might look like in terms of future ability to participate in trials is a critical thing to think about. Right,
and that can be hard to predict because that partly depends on how the next trial sets up their eligibility criteria. Yeah. And actually, and getting to the point of travel, yeah, absolutely. Yeah. And getting to Katie's point about travel is one thing to keep in mind is basically any study that involves travel, the sponsors of the research will pay for travel, so it's not something where you then have to fund your own travel. It's something that's reimbursed.
Yeah, and just thinking about also where your CF is, the benefits, the risks is always a consideration. So someone who's has a very stable disease, they don't appear to be progressing, they might be a little more conservative with what trials they want to get involved in, where somebody who has tried a lot of therapies, is still having their disease progress, might be willing to accept a little bit more risk for some of these novel therapies because there would be more benefit to them. So it's a very personal decision.
Yeah, I think the only thing I would add to that is for, my family, I think what you said about eligibility and whether it would conflict for other studies, I think I would want to pay attention to, like, what's the potential benefit of this therapy versus another, to weigh out, do I want to wait to try to enter this one versus this one? I think that would be key. So we want to miss the opportunity for a more impactful therapy, to participate in an earlier offer.
Got another question. The SpliceSense antisense oligonucleotide program seems to be the furthest along in development. Can you talk more about that therapy? We didn't talk much about individual mutations and the different types of mutations there are. I broke them into categories. What I know about the SpliceSense oligonucleotide or antisense oligonucleotide program, it's a lot easier to just say ASO. So their ASO program, as I understand it right now, is targeting one specific splice variant. That's a relatively common one. Does happen to be modulator eligible right now. That specific variant is a mouthful. It's 3849 plus 10 kb C to T. Those of you who have it will probably recognize that string of alphabet soup, but it'll be foreign to others of you. What matters with the ASOs is that they are all very specifically targeted towards the specific mutations. So meaning that trial that is targeting the drug that is targeting that mutation will only be eligible to people who have that mutation. And that while it could, if it's successful, could pave the way for other ASOs to target other variants, a new ASO would need to be developed for each variant that it would be intended to target. You can think of ASOs as kind of like little targeted magnets that zoom in and attach to the specific target on the DNA that they're designed to work towards. So they're very unique to the individual mutation that they're targeting. I don't know if anybody else has more to add to that.
Yeah, no, I think it really is just that specificity is the key point, that it's relevant for a specific subset. I think so far, actually, one challenge with that study is it's a mutation that's eligible for modulators, and I think currently being on a modulator makes somebody ineligible for the study. So it has to be somebody who both has the mutation and is not on a modulator. I don't know if that'll change in the future.
Another question, what have we learned from the research and approval of gene therapy for sickle cell disease? Additionally, I've heard that the sickle cell therapy requires chemotherapy. Would CF gene therapy also require this?
Yeah, I'm glad to talk about this a little. The sickle cell trials are a little... It's different when you have some treatments already existing for a disease. The process that you go through does look a little bit different. So I'm not sure there's much that we can learn from some of those trials that's applicable to CF. I can talk about the chemotherapy aspect. So sickle cell is a blood disorder. It involves the bone marrow, which has stem cells, which is why, yes, you have to go through a process to kill off some of these stem cells for the sickle cell therapy. That's not the same as CF. The cells we're targeting are not stem cells. There would not be this need to, kill off cells, do anything sort of similar to a chemotherapy for the types of gene therapies that we talked about today.
And I would say the way I would explain part of the reason for that is in sickle cell disease, there's an abnormality that's causing something bad to happen. So you need to get rid of that thing that's making something bad happen. And so that's why they need chemotherapy, is to get rid of the mutation that's causing harm. And then they give the gene therapy modified cells back to then produce normal bone marrow. Whereas in CF, it's just, you're just missing something good. You're not creating something that's causing harm. And so you don't need something like chemotherapy because you don't need to get rid of something bad. It's just trying to replace something good that's not there enough.
Another question. Mental health side effects have been associated with modulators. With more recent research, are we seeing any improvement in mental health? Is anything known about gene therapies and potential effects on mental health?
I think this is just a big area of focus for everybody who's involved in CF, whether both people who have CF and people who work in CF clinics. It's really, it's a huge concern. I think there's a lot of research that's going into trying to see if we can have improvements, see what we can do to improve mental health for people with CF, and just but partly just by treating mental health directly and by assessing what's the impact of new therapies as they come along. And if somebody's having severe mental health side effects of modulators, will other therapies give them the same benefit without the mental health side effect? I think it's too early to really say anything about whether there are mental health side effects of genetic-based therapies, because so far they've been given to so few people. But it's certainly something that everybody's going to be paying attention to.
And on sort of the theoretical side of it, one of the things we talked about was these gene therapies. Some of the early ones are inhaled, so they're going into the lungs. There's a chance that because we're targeting the lungs directly, you don't get sort of the full body exposure like you do with modulators. Maybe you'd have more targeted effects to the lungs than the brain. But again, like Dan said, so few people have had these. It is so early that we don't know. We can hypothesize, but until we have data, we just can't say for sure.
Another question. What are your thoughts on a timeline for an actual cure? I've heard everything from very soon to probably never. I've also heard some people say that a total one-time cure is unlikely, and maybe a once every 5 years or so is more likely for CF. I can start by saying I think cure means different things to different people. And I like that the person who submitted this question put cure in quotes. So I think that registers to that individual as well. You can talk about a cure for lung disease. You can talk about a cure for pancreatic disease. You can talk about a cure for CF-related infertility. And then you can talk about a cure meaning the concept of CF doesn't even exist. And I think there's different answers to all of those definitions of what cure means.
Yeah,
I don't know, Dan, if you have a comment on timeline.
Yeah, I would add to that too, that some people, a cure means I get a one-time treatment and I never have to treat anything again. And for other people, it's like, well, I do have to take something every day, but that makes me feel none of the effects of CF. So yeah, that's another different, a way that people might differentiate how they define cure. And I think, so that's part of the variability. And I think part of it is we just really don't know. And so anytime somebody's giving you their projection of what the likely timeline is for a cure, it's people giving their best guess. And I think it just kind of reveals, are you talking to somebody who's an optimist or a pessimist? And we don't know whether the optimists are right or the pessimists are right. We're just doing our best to figure it out as we go forward. And so it's, yeah, it is very hard to answer the question because we just really don't know until we get more information.
It's so hard that I'm not even going to take a stab at it because we don't know.
And I'm not qualified.
Here's another question. Would any genetic therapies be applicable for lung transplant recipients? Remember that the therapies we're talking about now are targeting the lungs. So if you get a transplant, you've got a new set of lungs that don't have CF. So the transport of the ions within those lungs is driven based on the donor's copy of CFTR or copies of CFTR. So these therapies wouldn't be needed in the lungs to target that. But as therapies, this kind of goes back to our last question in terms of what aspect are you trying to cure? As these therapies could potentially extend beyond the lungs, I could see there could potentially be a role there. Dan, you probably have something to add to that.
You know, I completely agree. I think the other thing that I wonder how it would impact things is, you know, anytime you're giving something that's a virus or any sort of vector, there's the question of what sort of inflammatory response there might be to it, and would that be altered when people are on immunosuppression for transplant? So I think that's something that is definitely pretty far down the road if that is something that's going to be approached, because I think there's so many difficult components to studying that.
Another question, is there any research to repair the reproductive system of men with CF? This isn't something I know about from a research standpoint in terms of adults. I can tell you there are case reports, and I've heard stories about infants who have had their reproductive system prevented from being damaged based on very early treatments. So I think it's early days for this kind of, for an answer that long-term looking. And that's not where genetic therapies are going to be targeted. I don't know of any active research in that area right now.
Yeah, I think it's a challenge because, you know, male infertility in CF is based on not having a vas deferens, and so it's hard to replace a vas deferens that's missing. So, but there have been even situations, case reports of women who are pregnant and find out that the baby they're carrying has CF, and then start taking a modulator because it's a mutation that's eligible for a modulator. And some evidence that some of those babies might be born with a functioning vas deferens or a functioning pancreas when they wouldn't have been otherwise. But yeah, again, that's something that's in the early stages as well.
How is CF genetic therapy research supporting research for other genetic conditions?
Yeah, I'm glad to tackle this one. In research in medicine, there's this concept called bench to bedside, and it's the idea that you have laboratory developments that ultimately end up with patients. But this is a back and forth flow. So it's not just how CF genetic therapy research is supporting other conditions. It's actually how we're learning from all the conditions about gene therapy, and also the basic gene therapy research we're learning applies to all other conditions. So sometimes there's a clinical trial that's done and they find, wow, this gene therapy delivery is a lot more effective if we did this to the viral vector before we used it on the patient. That can then be applied to other diseases, other areas of genetic research. So in short, everything we learn from genetic therapies in CF can apply to other diseases and vice versa. So this really is a very collaborative, from academic to the clinics environment, where everything we learn in one disease can and will be used in other diseases because it is such a fast moving and exciting landscape.
Another question, what research is underway for people with very rare mutations? For example, only a few people have it. Would they be eligible for the current genetic therapy trials in CF? Yes, that's the goal. Those are the people that will, although everyone could benefit from gene therapy if it's proven to be all of these genetic therapies, I should say, if and when they're proven to be functional. But those are the people that would be great candidates, as long as they're adults, for these genetic therapy trials, because they can't get a modulator right now and have the most to benefit from this type of therapy.
And part of it too is we talked specifically about the SpliceSense study, which focuses on one specific mutation. But I think it's important that when we're talking about things that are like replacing the DNA or replacing the messenger RNA, it doesn't matter what mutation the person has, because all we're doing is we're giving the normal version. So regardless of how rare your mutation is, you would be eligible for that.
And so in contrast to modulators, where you have to go through a long list and see if your mutations match the list of eligible mutations outside of the ASOs, which are very targeted toward those specific variants, it's open. You don't have to be on a list. You don't have to have a variant that anybody has ever had before. Is there any research into whether CF carriers could benefit from genetic therapies? I look at that as, is somebody truly a carrier, or do they have CF and only one mutation's been discovered? So the genetic therapies will be for people who have CF, and the definition truly of CF is not just based on genetic test results, but if you have clinical symptoms and other parameters of the disease. And so people who have proven CF, even if only one mutation has been identified, they would be eligible for genetic therapies. But people who are truly carriers, normal sweat chloride, truly just carriers, technically shouldn't have any symptoms, short of what appears to be a very small increased risk for pancreatitis. And so genetic therapies wouldn't be a good therapy for individuals because there's not anything to treat. Another question, would genetic therapies work for people with advanced lung disease? Dan, that's probably a good question for you.
Yeah, I think, again, this is something that is something we have to see the impact as we move forward. I would say that the best parallel we have is what we've seen with modulators. There's been a lot of variability of response, but we've seen some people who were, getting close to the point of needing lung transplant, who then their lung disease stabilized, and we can't guarantee that they'll never need a transplant, but it's pretty clear that we've gone from being close to being ready to list somebody for transplant to saying, hey, they've now been stable for several years and not considering listing for transplant now because they're too healthy for it. So the hope would be that we would see the same thing with genetic-based therapies, and time will tell as we move forward for these.
And I'll just add to that in terms of being a participant in a clinical trial would depend on the individual trial. Most trials are going to have specific requirements about a minimum FEV and other stages of disease in terms of, would you be a good candidate to be an active trial participant versus potentially receiving the drug after approval. So I think we are at time, but hopefully this was a helpful session where you got a little bit more comfort with talking about these terms and getting a little bit more used to the idea of genetic therapies. There are a lot of great resources out there. I know that there were slides in the chat and some links to some handouts, including one with a QR code to take you to the CF Foundation's resources on genetic therapies, which are a really good starting point. And please talk to your CF care teams if you have more questions. They'll be a good resource.